Hi everyone, Ray here again, this time with my third lecture in the series on the coronavirus. In the second lecture, I described to you how coronaviruses get inside of cells to take them over. In this lecture, it'll be like the last one. I'll divide it into two parts. In the first part, I'll describe how once the coronavirus RNA enters a cell, that RNA gets copied to make many more coronavirus genomes that can be packaged to make new coronaviruses that can then go on to additional cells, infect other cells. In the second part, I'll describe how the coronavirus proteins are produced and how those proteins interact with the RNA to assemble new coronaviruses that can then exit the cell and go on to infect other cells. Now, before I get started with the topical material, I'd like to give you two caveats. The first is that once you get inside the cell, things get a lot more complicated. As you'll see, a lot of proteins start getting involved. And we still only know partially what all these proteins do, and some of them we don't know at all. The second thing is that many of the studies were done on the SARS coronavirus 1, which was the cause of the SARS epidemic in 2003. They were not done on the SARS coronavirus 2. At least some of them have not been done yet because we've only known about that for a few months. Now, this is common in life sciences and biology studies that we study things in one organism and then transfer that knowledge to a different system. And so, for example, you can't do a lot of experiments on humans, and so instead we do experiments on mice, on fruit flies, on soil nematodes, even on yeast and bacteria. And because of evolution, what goes on inside the cell, even for a yeast cell, is very similar proteins and very similar functions to what go on inside of human cells. So it is with the coronavirus. Coronavirus 2 is very closely related to coronavirus 1. So even though there might be some differences in details, the general principles of what's going on are very, very similar. And so we can transfer a lot of the learnings we have from coronavirus 1, which we started as a field studying intensively after the 2003 epidemic, and apply that knowledge now to coronavirus 2. Okay, so let's resume where we left off last time. We have a cell, and I always draw the membrane in black here, and the RNA, which I always draw in red, has entered the cell. Now, the first thing that's going to happen is that RNA is going to be translated to make the viral proteins, and those viral proteins will be involved in copying that RNA. And that's done because the genome is an RNA, it's not a DNA, so you can translate it right away. And that's done by the ribosome. So the ribosome is essentially a protein-making factory inside the cell. And you can think of it like a 3D printer. Like in a 3D printer, you have a computer file you send to the printer, and then the printer makes a 3D shape out of it. In the same way, here, the computer file for the ribosome is the RNA of the coronavirus, and the shapes are the proteins that the ribosome produces from the RNA. And so I'll draw some ribosomes here on the coronavirus mRNA. And these are very simplified versions of what a ribosome looks like. And then emerging from the ribosomes are going to start here and go in this direction. So you have a little bit of protein coming out of this one, a little bit more out of here, and a little bit more yet out of here. So there's two very long RNAs that are produced when the two very long proteins that are produced when the coronavirus RNA enters the cell. And those proteins fold up into different shapes. And I'll draw those shapes out here. So let's imagine we have a triangle, a pair of scissors, a square, another pair of scissors, a circle, etc. And these are all linked together. So this is one very long protein. Now, I've already described that there are molecular scissors that work outside the cell to clip the spike protein. Similarly, these molecular scissors can cut these linkages here to release these individual shapes. So we end up with a triangle, we end up with a couple pair of scissors, we end up with the square, and we end up with the circle as separate proteins. From the longer of the two proteins that are made from this RNA initially, 16 different proteins, just wrap your head around that, are produced and released to make these individual structures. Now, I'd like to say a little bit more about proteins. I've drawn them here as shapes, and that's actually very apt because all proteins have a characteristic shape. And that shape enables them to do their job in much the same way that everyday tools that we use have very specific shapes that enable them to do their jobs. And some of those tools, beyond having a shape, can move. So here's an example, a pair of pliers. It moves, that's essential to its function. Similarly, there are proteins that, like pliers, can open up and grab things. There's another class of proteins known as regulatory proteins that control things, they regulate things. Well, here's a gate valve. This also regulates something. It moves to open up a valve and it regulates the flow of water. In a similar way, there's proteins in the cell that regulate the flow of information, for example. Another example is we have a hammer here. This doesn't have any fancy moving parts in it, but it illustrates another principle in that it bangs a nail in and it can pull a nail out. So it's multifunctional, it has two functions. There's a lot of proteins like that are multifunctional. They could have 2 functions, 3 functions, 4 functions, etc. And you have a very simple tool like a screwdriver, but again, its shape is key to its function. So you have different tools to do different things. They all have their characteristic shapes, and likewise, the virus makes different proteins with different shapes, each of which is specialized to do something specific. Now, to replicate this RNA, so let's take the RNA and show it up here. There's a group of 5 of these proteins, 5 out of the 16, that come together and in sort of an assembly line fashion, they work on this RNA to help make copies of this RNA. Now, this RNA is what's known as a positive strand RNA. There's one strand, it's single-stranded, and if you recall, it has a sequence of nucleotides like A, G, C, and U instead of T. So if you recall from DNA, let me draw DNA here quickly. So DNA, you have bases that, DNA is double-stranded, first of all. You have 2 different strands, and they're linked by bases bonding to each other. And so some of you may recall that of the 4 bases, you have A is always bonding to T, in RNA that would be U, and G is always bonding to C. Now, let's take this RNA and imagine there's a sequence here of A, C, G, U, U, C, A. Okay? Now, to copy this, all you would do is you'd put in the complementary base using these rules here. So this would be U, this would be G, this would be A, this would be A, this would be C, this would be G, and this would be U. And you keep going, the whole RNA is copied that way. So 30,000, all 30,000 roughly bases are copied to produce what's called the minus strand RNA. Now, even though RNA is usually single-stranded, it can be double-stranded, just like DNA is. And so once you've copied it, now you have this double-stranded RNA. Well, this uRNA can be taken over here, and then it can be copied. And it gets copied many times. And every time you copy it, you're making a plus strand. So, you copy it the first time, you make 1 plus strand. The second time, you make another plus strand. The third time, you make another plus strand, and so on and so forth, until you've made many, many plus strands, and those can then be used to produce new viruses.